All right, I'm excited for this topic because I, I mean, I know everybody now associates viruses with COVID, but once upon a time, before we were all wearing masks and freaking out, I worked in a lab with HIV, and that's like where I fell in love with research, and so I love this topic. Anyways, so viruses are infectious agents that are not alive, and the reason they're not alive is because they can't sustain themselves without a host. They absolutely need a host in order to live. All they are, they're basically genetic material, maybe some enzymes, in a shell. And that shell can be like a capsid, or it can be a capsid and an envelope. And a capsid is just this outer coating made of proteins. And these proteins go in units called capsomeres. And they usually make like a pretty geometric pattern. And you can see some of them here. But anyways, so a virus is just going to be those things. There's no ATP, there's no DNA polymerase. So they're missing a lot of stuff that you need in order to be classified as a living thing. They can't reproduce on their own. They can't metabolize anything on their own. So we're going to see how exactly they hijack the hosts in order to be able to do all this stuff and in order to make more of themselves. So here are some examples of viruses. They're actually quite diverse. They don't just infect humans or even eukaryotes. They also infect bacteria. There are viruses that infect plants. But let's dive into how they work. So this is an example of a DNA virus. There are DNA viruses and there are RNA viruses, and we'll go into RNA viruses afterwards. So first we're going to look at DNA. And DNA viruses, they don't even need any proteins at all in them. Or not, when I say protein, I mean like enzymes. Like they don't need their own polymerase or anything like that. So what they're going to do is they have on their capsid, the virus is going to have some type of protein or glycoprotein. And this protein has a specific shape or conformation, and the host cell has a specific receptor with a matching conformation. And this receptor was maybe once beneficial to the host, but now viruses have taken advantage of it. So these two are going to bind very specifically, and the virus is going to convince the cell to let it in, to endocytose it. And I kind of like to think of this like a fake ID. So the virus can tell the cell, I'm something good, I want to get in, and the cell will let it in. And in comes the DNA, and that DNA is going to be incorporated into the host genome. And once it's in the host genome, then the host can use all of its DNA polymerase to replicate it, all of its mismatch repair systems to make sure that there's no mutations going on as the cell divides, and then it can use its whole transcription and translation machinery to get these viral proteins going again, and these viruses can self-assemble spontaneously on their own. Once all these components have been made by the host, they just assemble, and then they break out of the cell and they infect more cells. So that's a DNA virus. There's also RNA viruses. RNA viruses tend to be enveloped. This is what HIV is. HIV is more complicated. We'll go into HIV later. This is essentially what the SARS-CoV-2, the virus that causes COVID-19, does and what it looks like. It's an RNA virus. RNA viruses are going to have their own polymerases. They have to have their own polymerases because no host is going to have a polymerase that functions to make RNA into more RNA. And that's essentially what this virus has to do in order to make more of itself. So if it's got RNA, and if it's got polymerases that can turn RNA into RNA, then the virus can have more RNA, which the host cell, can then use its machinery to make that into protein. So there's going to be pretty much the same mechanism of entry. You're going to have some kind of specific protein on the outside, and the cell is going to have some kind of specific receptor. Except this time, instead of the cell endocytosing this virus, the virus can actually just fuse its envelope with the membrane. It's like a vesicle, but it's outside the cell and it's got evil stuff in it, so it can just fuse with the membrane and come on into the cell. And once it's inside the cell, the RNA polymerase of the virus can replicate its RNA, and then the cell is going to make all that RNA into viral proteins, and the viruses are going to self-assemble the way that they had been doing. So I said we were going to go into HIV specifically, and the reason why is because HIV is a bit of a different RNA virus. Instead of having an RNA polymerase that makes RNA to RNA, HIV has something called reverse transcriptase. HIV is a retrovirus, and retrovirus is this whole class of viruses that does this. Viruses that have RNA, and they're going to have a reverse transcriptase that makes this RNA into DNA. And this DNA can then be incorporated into the host genome. And then the host can use its machinery to make all the viral components. And what makes HIV so lethal is the fact that it's going to infect your immune system, so you can't defend yourself against this virus or any other infection that happens to come your way after you get HIV. So HIV specifically infects helper T cells in the immune system. So those are the cells that alert your other specific adaptive immunity cells about what they need to do and when they should be active. And these helper T cells have a receptor on them called CD4. And CD4 is going to be what HIV binds to. HIV's got this glycoprotein on its envelope called GP120, and it's got another one called GP41 that's going to kind of help out. And GP120 and GP41 are going to bind CD4 and also this other accessory protein called CCR5. And that's going to convince the cell to let this virus fuse with the membrane and enter the cell. Once inside the cell, this RNA inside of HIV can be reverse transcribed by the virus's own reverse transcriptase enzyme into DNA, and this DNA can then be incorporated into the host genome. And now the host pretty much has to make all these viral components for HIV so that it can make more of itself and exit the cell and continue to attack more immune cells. So that's how that works. And then the other thing that I think is really interesting to discuss is viruses that infect bacteria. They're a whole different thing. They're called bacteriophages. You'll often hear them just called phages. And so these phages, instead of getting their whole self into the bacterial cell, they're just going to inject their DNA on the outside, from the outside into the inside. And they kind of go through two cycles. There's the lytic cycle and the lysogenic cycle. So we're first looking at just a lytic cycle. And you can see why it's called lytic. Lysis is when a cell breaks or bursts. So the lytic cycle ends with basically death for this bacterium. So this phage is first going to attach to the outside. It's got lots of little appendages on its bottom that it can use to attach to the bacteria. And it's going to inject its DNA into the bacterium. That DNA is going to hang out in the cell until the bacterium has made all the viral components out of the DNA through transcription and translation. And then those viruses are going to self-assemble just like the other ones we looked at for eukaryotic cells. And they're going to burst out of the cell and kill it and go and infect more cells. This is one type of phage. Other types of phages have two cycles, the lytic and the lysogenic cycle. The lysogenic cycle is basically a way for the virus to lie dormant and get itself infected into more cells. So occasionally what will happen after the initial infection, after insertion of this DNA into the bacterial genome, is if this DNA stays in the genome, nothing's going to happen. The bacterium's just going to live its little happy life and divide a bunch and make a bunch of progeny that also have the viral DNA, by the way. And the trouble comes when sometimes DNA in bacteria will not be in the genome. It will be in these little units called plasmids, which are like tiny circles of DNA. And sometimes the viral DNA will escape the genome, and you can see that here. And if that happens, then some chemical stuff happens inside the cell, and the cell decides if there's going to be a lytic or lysogenic cycle. And if the lysogenic cycle occurs, basically nothing happens. DNA goes back into the genome, and we make more progeny that also have this viral DNA in the genome. If the lytic cycle is triggered, then what happens is basically what we saw before, where these phages get assembled because the bacterium is going to make all the proteins, and then they're going to basically just come together all by themselves and burst out of the cell and kill the cell. So that's a pretty interesting mechanism to look at. And phages are cool because they helped us with a lot of discoveries. So actually there were these two experimenters called Hershey and Chase back when they were trying to figure out what was the genetic material. How do we pass things down? Was it through RNA? Was it through protein? Was it through sugars? Was it through lipids? We didn't know. And they took a bacteriophage and they radio-labeled just its DNA. And then they put it with bacteria, and they saw what actually ended up inside the bacterium by detecting the radioactivity. And it was the DNA. And that's one of the experiments that gave us evidence that DNA is the genetic material. There's one other infectious agent I want to look at, and it's not a virus. There are these infectious proteins, and they're called prions. And they have caused some trouble for us humans. But the way that they work is that in some way, this prion, this infectious protein, when it bumps into another protein, it makes that protein change its conformation or shape in order to look like the prion. It won't change its sequence, it'll just change like the secondary structure specifically. So prions have beta sheets. You might recall from learning about protein structure that there's two types of secondary structure. There's alpha helices, which look like this, and beta sheets that look more like this. And something about the way that the prions, which are beta sheets, are going to interact with other proteins that are alpha helices can turn those alpha helices into beta sheets. And if enough of those happen, these prions can build up plaques. And this has happened with proteins in people's brains, and it's caused a lot of disease. So this can't cross organisms. It's not like two people can be in a room and one has a prion and the other one is afraid of being infected. It's going to be within one multicellular organism.